For my project, I studied the paper called cadherin gene alterations are rare events in thyroid tumors. The reason I chose this paper was because the thyroid is one of my favorite topics, ever since I learned about it in physiology and endocrinology. It's interesting for me to see how cell mechanics, something I learned about in this class, relates to a gland that I've always learned to be controlled by hormones. As we learned, cadherins are a type of cell-adhesion molecule that play a role in forming adherent junctions which bind cells to each other. These are calcium-dependent transmembrane glycoproteins that have tandem-repeated sequences. These are called cadherin domains that are located in the extracellular domain. The function of cadherins depends on the cytoplasmic binding domain. This domain connects the cadherins to the cytoskeleton through catenins. As you can see in the picture, it connects it to actin fold. Cadherins are known as the epithelial cadherins and are on the surface of one cell and can bind with others of the same kind to form bridges. If there is a loss of cadherin, it is usually involved in the formation of epithelial types of cancers such as carcinoma. These changes are particularly prominent in poorly differentiated, undifferentiated, and/or invasive tumors. This loss of expression of cadherin has been associated with the mutation of the cadherin gene and loss of heterozygosity. Cadherins are sort of like binder tabs. They keep the papers in the right place and properly in the right order connect to the rest. But if one binder tab were to fall out, then the papers would keep stacking up on one another and not properly fit in the right place. The focus of the paper was to understand and analyze the expression of cadherin in differentiated malignant thyroid tumors. They wanted to know if this is related to irreversible abnormalities like loss of heterozygosity or mutations, or structural abnormalities in the cadherin gene. This brings up the question, why thyroid? Thyroid cancers are poorly understood in science. Researchers don't know if the cause of thyroid cancers is genetic, environmental, or both. Previous thyroid studies have shown some or no expression of cadherin, so researchers wanted to further explore this. The papillary carcinoma is one type of thyroid cancer. It is the most common, slow-growing, differentiated cancer that develops in follicular cells. It can develop in both lobes of the thyroid gland. It can also spread to nearby lymph nodes, but is overall treatable. Follicular carcinoma is the second most common. It is found when there is not enough iodine intake. It's a differentiated cancer and a little bit more aggressive than papillary and can spread to other organs. Adenomas are benign. There are clusters of cells that are not cancerous and will not spread to other areas. And finally, differentiated cancers look more like normal cells, while undifferentiated are immature cells that are hard to recognize and are thus more malignant. Researchers took 4 types of tumor specimens and used immunohistochemistry, PCR, sequencing, and loss of heterozygosity to understand the expression of cadherins in thyroid tumors.
Can I actually ask a question about the last, like one more back, Simran? I don't really know anything about the difference between differentiated versus undifferentiated cancer tumors, really. Like, I guess, like, would an undifferentiated cancer cell, like, is that like worse, or, like, because it's more mobile? That because epithelial to mesenchymal transition, it like is becoming like less. I mean, it's not less differentiated there, but it is differently differentiated. So yeah, that's the question.
What I got out of it was that it's just harder to recognize it and understand its mechanism just because it's not developed or even close to being like recognizing what type of cell it is. So I guess that's why it would be a little bit worse than differentiated. But
like it's harder for like the immune system to capture it. Is that?
Just like if you take a look at a differentiated cell and an undifferentiated cell, it's harder to like recognize what type of cell it's becoming, the undifferentiated one.
I see. Like what type of, okay. All right.
Yeah.
Gotcha. Thank you.
Researchers took 4 types of tumor specimens and used immunohistochemistry, PCR, sequencing, and loss of heterozygosity to understand the expression of cadherins in thyroid tumors. To better understand the immunohistochemistry results and figures, I created a model that shows the cell types for different thyroid tumors. The key shows that pink indicates an intense cadherin immunoreactivity, orange indicates a less intense immunoreactivity, and yellow indicates a faint reaction. If you look at the lymphocytic side of the cell, you can see that there is more membrane immunoreactivity. The pink color of the lateral border of the cells shows that there is more intense staining here than the basal border of the membrane, which is colored orange, and even more intense staining than the apical border, which is colored yellow.
I'm sorry, Simran. Could you play the last slide in this one again? There was some audio coming through and I couldn't hear what was going on. Thank you.
To better understand the immunohistochemistry results and figures, I created a model that shows the cell types for different thyroid tumors. The key shows that pink indicates an intense cadherin immunoreactivity, orange indicates a less intense immunoreactivity, and yellow indicates a faint or absent immunoreactivity.
Can I ask another question? So is this, is this like bean shape? Is that the cell? Or is that
Yeah.
the new? Okay, so then what's the square?
It's just like a close-up of the cell,
Okay.
like what you in the microscope. Yeah, you can tell the first square, I kind of labeled it with the cytoplasm and membrane.
Yeah, so does it have that intense, I was confused about that intense immunoreactivity. Is there pink like intensity at those edges?
It's just indicating that there's another cell there.
Okay.
So, yeah.
Okay.
So the lateral membrane would have intense immunoreactivity.
I see.
Sorry. No, you're fine. I was just waiting for the, I probably have to wait 2 more minutes.
I'm not sure who that is, but yeah. We can hear people in the background of somebody's thing. Okay. Let's keep rolling. Sorry.
Okay.
If you look at the
It's
normal
Aaron.
thyroid goiter and lymphocytic thyroid cell, you can see that there is more membrane immunoreactivity. The pink color of the lateral border of the cells shows that there is more intense staining here than the basal border of the membrane, which is colored orange, and even more intense staining than the apical border, which is colored yellow. Follicular adenoma also had similar results, but in addition to the membrane immunoreactivity, it also had cytoplasmic staining. The significance of this model and the results is that both of these non-cancerous types of thyroid cells show a polarized expression of cadherin. This is seen by the uniform pattern of colors shown in this model. Next, for follicular carcinomas, strong to moderate immunoreactivity was homogeneous throughout the tumor. You can tell this by the pink and orange randomly around the membrane. Some cytoplasmic staining was also observed, and you can see this by the pink and orange in the cytoplasm. What makes this significant is that this is not the typical polarized pattern you see in normal thyroid cells that we saw in the last slide. This is non-polarized cadherin expression. For papillary carcinomas, immunoreactivity was generally less than in follicular carcinomas we saw on the last slide. You can see this by the orange color. Also, the immunoreactivity was heterogeneous, meaning that positive areas coexisted side by side with negative areas. You can see this by the gaps of orange around the membrane. These carcinomas had more strong cytoplasmic staining. You can see some added pink in the cytoplasm.
Simran, can I jump in here?
Yeah.
Because I want to help us out. So What's going on here? So the reason why I was confused about our figure, like, because we have this square and then this bean shape in the center, is because what we're actually looking at. So are you interpreting this as a single cell, this micrograph here? Like kind of like this like dog shaped thing in the center is like a single cell or
Yeah.
something? Yeah, so what we're actually looking at here is a tissue histology slide.
Or
So
actually, a cell would be like a zoom, version. So that would be like a bunch of different cells. Yeah.
yeah, so this is full of cells. Each one of these like dark areas is like it's like the membrane around the nucleus essentially is what it looks like. Can you go back to the previous slide?
Yeah.
So like around that hole, that lumen there, each
Mhm.
one of those dark circles is a nucleus, and then the cadherin staining is, those little projections coming off of it towards
Okay.
the center. Does that make sense? They look like little antenna coming off of those those
Yeah.
nuclei. Does that make sense? So what you've actually done here is you've made a kind of like cadherin map of like where it's really strong at the tissue level though.
Yeah.
Which is cool. But it's not at the cell, it's not at the cellular level. You're making like a tissue topology of that signal, which is really cool. Are we on the same page here with what
Yeah.
we're looking at? Cool. All right. Thank you. I thought you were talking about like a single cell in the model.
Yeah. Oh, yeah. You're right. It is a.
Yeah. Okay. Cool. And it doesn't really matter if that's how you interpreted it, because like that's cool that you did that, but you still made this sweet map of the data. It's just at a tissue level. Does that make sense?
Yeah.
Cool. Awesome.
The significance of this carcinoma.
Sorry. Let me also mute myself.
The significance of this carcinoma was that it was also non-polarized in its expression of cadherin throughout the cell. For the poorly differentiated carcinomas, the scattered pink and yellow show some areas had strong membrane immunoreactivity, while less differentiated areas had faint immunoreactivity. Again, the main point is that cadherin's expression was non-polarized. So what does this mean? The results
Can we just, I think that's really striking. Can we just I would just like to appreciate that picture a little bit more. So can everyone appreciate that, how that cadherin is all the way around the membranes now? Like you can really see it around all of those membranes. And can you go back, Simran, to like one of the previous slides? So we can just look at that. The one more would be good.
One more back.
Yeah, one more back. Like, it only has, we don't have that cadherin down at the base or even really basolateral. It's not below the nucleus. It is in some cases. We can't really see the outlines of those cells other than at those two little side projections, not even on the apical surface. And then let's go forward again. One more. Right there. Yeah. So then you can see it all, you can see the outlines of a lot of those cells. It's really nice. Cool. Did they quantify that at all?
Not really.
Okay.
They just explained that it was where it was intense or not, but no, they didn't say.
Cool. Awesome. All right, sorry.
So what does this mean? The results we found for our papillary carcinoma confirmed results of other studies mentioned in this article. The results support Edelman's study, who found that non-polarized expression can be observed in carcinomas with histologic structures like papillae in our papillary carcinoma. We also see a pericellular redistribution of cadherin, just like Serini's study, whose results stated that this redistribution might be due to a decreased tyrosine phosphorylation of cadherin cytodomain. The low and heterogeneous expression of papillary carcinoma also confirms Braben's study, who observed a reduction in E. cadherin mRNA in papillary carcinomas. In one of the many cases of papillary carcinomas, there was a shift that could be attributed to a missense mutation, changing alanine to threonine. The substitution apparently didn't interfere with the recognition site of the antibody and did not change immunoreactivity. Thus, this case was no different than the rest of the papillary carcinomas. There was also no evidence of the loss of heterozygosity in differentiated carcinomas. For the poorly differentiated carcinomas, we didn't have enough to conclude the general occurrence of loss of heterozygosity or mutations. Our results show that both loss of heterozygosity and mutations at the E. cadherin locus are rare, thus leading to the conclusion that the downregulated expression of E. cadherin cannot be ascribed to loss of heterozygosity nor mutations. This study supported the results of the other studies about the non-polarized expression of E. cadherin in thyroid carcinomas and that mutations and loss of heterozygosity is rare. It leads them to support that there is some sort of transcriptional regulation that creates morphological changes in E. cadherin. In support with researchers Shiozaki and Serini, the paper suggests that if these cells are treated with an epidermal growth factor, then the epidermal growth factor receptor could be a candidate kinase that phosphorylates tyrosine and impairs E. cadherin. Shiozaki also reported that after treatment with epidural growth factor, thyroid cell lines of papillary and follicular carcinomas had enhanced migration and the tumorigenic capacity became higher. For the discussion activity, brainstorm and come up with an analogy that is analogous to how post-translational modifications like EGF can work in a tumor thyroid cell.
So you can put them in breakout rooms, or if everyone just wants to brainstorm and share. You're muted.
Let's do breakout rooms, and because we already have it in pairs, so that should be nice and easy. And just can you repeat the question for us, and then we'll have opportunity for questions about the question, and then we'll open up the rooms.
For the discussion activity, brainstorm and come up with an analogy that is analogous to how post-translational modifications like EGF can work in a tumor thyroid cell.
Okay, so post-translational modification is basically just slapping something onto a protein, like adding a sugar to it or a phosphate or ubiquitin or biotin. What are other things you can slap on a protein? I don't know. Probably put nucleotide on there. What else? So how might that affect the tumor if we have protein modification? Is that what you're asking?
Yeah, exactly.
Okay, so yeah, let's just come up with different ways, and let's come up with different modifications too, like I just did. You could use one of those too. How might ubiquitination, for example, affect a cancer cell if you're ubiquitinating something? And if you don't know what the frick that is, it's okay. Just talk about what you know. All right, I'll open up the rooms and have fun. Oh, Cody is unassigned. Cody, you can just hang out with me and we'll discuss it. So everyone should be able to hang out in their room. I think Simran, I paired you up with. Actually, let me move Cody to your room into breakout room 1. And Simran, you don't have to do the activity because you're the lecturer. So how's that sound? And actually, Kate is alone right now because Mina's not joined. Let me move Aaron with Kate and they can discuss. Oh no, never mind. Well, yeah, I'll let those three go hang out together.
For the results, I'll go back to the analysis.
Okay, how long do you want me to keep them locked away? Simran? Are you there?
Tori, did you say something? I think you were muted.
Yes. I'm
I
muted?
can't hear you still. I don't know why.
You can't? It says I'm talking. I'll be back. Hey, can you guys hear me?
Yes, we can hear you.
Okay, something's wrong with Simran's speaker or something. Okay.
No worries. We were just saying that maybe some modifications to some proteins on the plasma membrane could inhibit our immune system from like targeting the cancer cells.
Ooh, yeah. Like some kind of like capsid or something around the cell, like a protein to like protect it.
Mm-hmm.
Oh, I was
From
actually
infection.
thinking more
Oh, yeah.
like certain modifications will cause a protein to get trafficked to the cell membrane.
Yeah.
So if you lose those modifications, then like if it's like the tumor marker protein or something, if it's not getting expressed in the cell membrane anymore, then it's not.
Right. Yeah, so if we like modify those so that then they're getting degraded.
Yeah.
Yeah, that like if we're ubiquitinating a bunch of like receptors that like the immune system would recognize to hide those and sequester those. Yeah, that's a way easier way. I was thinking about manufacturing a capsid of like biotin
Yeah.
or something, which I mean, that's what like LPS is, lipopolysaccharide. That's a polysaccharide modification on a lipid, and it makes this huge, basically dense, lush, like carpet on
On the whole
bacteria.
cell.
Yeah, on bacteria. The E.
Wow.
coli is covered in LPS, lipopolysaccharide, and that's what actually makes you sick. Is your body detecting that and being like, no, we got a bunch of LPS in here. That means we got bacteria. Get it out.
Oh, I didn't even know. That's pretty cool. I didn't know that.
Yeah, so that's what your body actually detects, and it protects the bacteria because it's almost like a mucus layer because it's just
Yeah,
sugar, so it holds onto a bunch of water, so it's like a gel coating around the bacteria, and it's like very like
Interesting.
if I was like about the size, if I was about half the size of a bacteria, I would live on a bacteria because it'd be so comfortable. It'd be so squishy and soft. Okay.
That's fine. How about that's what we.
Okay, I'll let you lead the discussion for us, Simran.
Okay. Does anybody have any examples that they came up with?
I didn't think of a specific one, but I was just thinking of like if you mark something for degradation with ubiquitination, just something that controls the cell cycle, so that way the cell cycle keeps going instead of being regulated, that could contribute to a tumor.
Mm-hmm. Did you come up with an analogy for that? Like to real life?
See, I can't really think of one necessarily, but just like, I guess if you add something that doesn't make like the brakes to your bike work anymore, then it's harder to stop it.
Yeah.
But I don't know what necessarily that would be.
Yeah, like a malfunctioning brake in a car or bike.
Yeah.
Well, one of the things that we talked about was adding a pull-up bar to like your door, like, and then the door can no longer close, so it's being like blocked. The activity of closing a door.
Yeah, that's a good one. Anybody else?
I came up with one.
Yeah.
I'll let other people go though, but I have one for that.
Okay. Does anyone else have one?
Has anyone ever seen Fern Gully?
No.
I need people to speak up if you've seen Fern Gully. Whoop whoop. Oh, whoop. Have you seen Fern Gully? Caw-caw! No, no one's ever seen Fern Gully. Okay, so Fern Gully is the best Disney movie that's not a Disney movie. And in Fern Gully, it's basically, who's seen Avatar, the Steven Spielberg film Avatar?
Yes.
Several
Is that the
people
one
say
with the
it.
blue people?
Yeah, that's the blue people in the
I've seen
forest.
Avatar.
It's the blue people in the forest, and like, it's all about nature and all that stuff. That's basically Fern Gully. Avatar stole Fern Gully swag. They stole their stilo, but I'm trying to share my screen. Anyway, in Fern Gully, it's about deforestation and stuff like that, just like in Avatar. And they X the trees. That's a modification of the tree, and that says, okay, break this one down, and that's like ubiquitination. So by like removing, who was it? Yeah, Hamza. That kind of goes off of like, did you share yours, and who was grouped up with you? I can't remember.
Oh yeah, it was me and Georgie, I think.
Georgie, yeah. Yeah. So do you want the ubiquitination and all that?
Yeah, so we were sort of thinking like there are certain tumor sort of marker proteins that get expressed on the cell membrane. So if there are modifications that would cause those proteins no longer be expressed, so if they get ubiquitinated, or if like the signals within the cell that cause the protein to get trafficked to the cell membrane are removed, then you sort of lose those signals and it's less likely to get attacked by the immune system.
Right, like ubiquitination is the system that the cell uses to mark things for destruction. But like in deforestation and in logging, like you just mark the tree with an X or whatever. Like if the tree's diseased in a forest or something, you go through and you mark them all with an X, and that's how you know which ones to destroy. So instead of
Tori, I just want to let you know that I have seen this movie now that you pull up this like tree with the X on it.
Yes, Georgie.
I remember.
I've seen it too.
Who? Yes. All right. Fern Gully. This is the Disney movie that I like. This is my favorite Disney movie, and it's not a Disney movie. Cool. All right. Stop. Sure. Cool. Fun activity. All right. Let's go back. Simran, you can have the stage again.
For the results, I'll go back to the analogy of the binder tab. Normal cells can be seen as binder tabs that organize papers. Tumor cells can be seen as binder tabs that are broken or missing, thus leading to a disorganized binder and papers stacking up on each other. It is not likely that the tab just fell out or broke, just like a loss of heterozygosity or mutation would explain. It is likely that something is wrong in the tab itself when it was manufactured that led to its breakage. This could be compared to the epidermal growth factor that phosphorylates tyrosine and thus leads to suppression of cadherin itself.
I think that analogy is really interesting. I like that a lot. And like like you said, it's unlikely that the binder tabs would fall out over time. But like that is like something that totally can happen too. Just like, our DNA and mutations and things just accumulate. Those insults is like what you call them. Those insults accumulate over time. And like, this is such a good demonstration of that. Those same insults happen to things like paper and file systems. And that's cool.
Yeah, the article kept saying that it's like really rare for it to happen.
Yeah.
So that's like unlikely for it to fall. But
yeah,
again,
in the DNA world, it's very unlikely.
Yeah.
It's also unlikely in this world too, but like if you give it enough abuse, it definitely can happen, right? Yeah. It's cool.
In conclusion, the paper supports that irreversible alterations of the cadherin gene are rare and that non-polarized expression likely reflects the transcriptional regulation or post-transcriptional modulation rather than structural abnormalities. The paper suggests that morphological changes might be due to an epidermal growth factor, which can cause a change in the localization of cadherin from cell borders to the cell surface. This study mostly focused on the immunoreactivity of cadherin expression in different thyroid tumors. For future directions, I would focus more on getting evidence on post-transcriptional modulations like the epidermal growth factor, rather than relying on other studies for evidence. I would also include a greater number of specimens. This study only included 31 tumor specimens.
What were the tumor sources? They're human, right?
Yes, I'm pretty sure.
Cool.
Thanks for listening.
Yeah, that was it.